5 days ago, I tested positive for SARS-CoV-2. Since that day, I've been isolated in this room, but I'm glad that I'm able to speak at this venue. And as you can see, it's a mild case, and that's very likely because I'm fully vaccinated. And so, though it's a breakthrough infection, it's mild as expected. The vaccine is doing its job. The development of the COVID vaccine has to rank among the greatest scientific accomplishments of all time. And if you think about it, it's a showcase of the power of genomic knowledge. The sequencing of the SARS-CoV-2 genome, which happened very soon after the virus actually emerged, led to the development of very specific tests, like the test that I used to test positive, and the development of these vaccines, which are now available globally and is protecting me and people around me. So really, I had wanted to talk about biological information, and now this example of the vaccine is something which I think is worth starting with to say how enormously powerful it is that we're living in this genomic era. But what I actually wanted to talk about when I first accepted doing this talk was about the limits of biological information. And so that's really the beginning of my talk here. Just a couple of weeks ago, a company made the news raising money with the goal of wanting to de-extinct woolly mammoths. Now, setting aside whether you think this is a good idea or not, let's just discuss, what it actually means to take, let's say, a mammoth genome and try and create a living, breathing mammoth from that information. And this will lead us down the path to really understanding in greater depth what biological information means and what metaphors we can use to understand it. So first of all, I think biological information these days is common currency. In the digital era, all of you are digital natives. We can actually translate between biological and digital information very easily. So the SARS-CoV-2 RNA genome, for example, it's about 30,000 bases. It's about 10 KB of data. The E. coli genome, it's about 4 million bases. It's about 1 MB of data. And the woolly mammoth genome, which is roughly the same size as our genome, it's 4 billion bases. And it's roughly a GB of data. And if you like to think of it differently, SARS-CoV-2 is like a small image. E. coli is like a music album. And woolly mammoth is like a proper HD movie on a DVD. So this sort of correlates with the idea that more complex species have bigger genomes. But you should be thinking that something is a bit mysterious here. So the question of de-extinction, is it really possible to de-extinct a mammoth if we have access to a complete woolly mammoth genome? Right? Is it really possible? And if you just think about this for a couple of minutes, you'll start to realize it really doesn't make sense that the number of bits needed to encode a living, breathing mammoth is the same as the number of bits needed to encode a movie about a mammoth, right? Something is off in this calculation. There's some missing information somewhere, and the goal of this talk is to see if we can get at resolving this paradox. So the start of the resolution is to understand that just like a DVD needs a DVD player or your thumb drive needs some sort of device to read it, genomic information on DNA or RNA is meaningless without a reader. It's meaningless without a device to read and execute what's on that information. So as a thought experiment, suppose we could have a universal genomic reader, right? So I have a machine where I have a tube of salts, sugar, water on one side. I pipe it into this black box, which I call the life machine. I put in some genome, and what comes out is living organisms. It seems, astonishing. It seems like science fiction. But actually, it's not so crazy, right? Because this is exactly what's done in any microbiology lab every day. You take salt, sugar, and water. You put it into another tube, which contains a single bacterial cell, which contains its own genome, of course. So this is bacterium A. And that bacterium A will make more copies of bacterium A. So this is something which, doesn't seem surprising anymore once I've revealed what's in the box. Actually, it should be surprising. We're just conditioned to think of this as not surprising. But actually, it's a fairly astonishing thing that a cell can take molecules and make other copies of cells. But setting that aside, what we do have here is a machine that can take a genome and make more copies of that cell, assuming you start with a cell. So Craig Venter actually showed several years ago now that the following thing was also possible. You could take bacterium species 1, which is this red cell, and use that as a reader for the genome of bacterium species 2. And if you do that, what you get out is actually cells of bacterium species 2. So in other words, it is possible for a cell to read the genome of a closely related species and carry out the instructions in that gene. But before you get too excited, this is obviously not possible. Okay, you can't take a woolly mammoth genome, shove it into a bacterial cell, forget the fact it wouldn't fit and all that. But even if you could fit it, you wouldn't get out a woolly mammoth. And that's not because a woolly mammoth is multicellular and needs to be gestated. No, you won't even get a woolly mammoth cell, right? Because somehow a bacterial cell is not capable of executing the information on a woolly mammoth genome. Okay, now I'm just going to step aside for 1 second and say that the following thing is possible. You could take reader 1, which is that red cell on the top left, put in genome 2, the green one, and make cells of type 2. Then you could put in genome 3 and make cells of type 3. And as long as at every step you're careful to make only small changes in the genome, you could keep doing this for a long time. And in the end, you might well get a woolly mammoth, or at least a woolly mammoth cell. How do I know this is possible? Because it's called evolution. This actually happened, right? I mean, the downside, of course, is that it could take billions of years. Still, it's possible. As long as every reader and every genome, you only make incremental steps. So the point I'm making here is just that, like on your smartphones when you upgrade the OS, right? An app doesn't work across different versions of the OS or on different kinds of devices. So the genomic information can only be decoded by a compatible reader. There's no such thing as a universal biological reader. So when we come back to the main question about de-extinction based only on a genome, to make a living organism from its genome, you also need the reader. The reader, it's a cell of the same or a closely related species. Now, suppose you didn't have a cell, you just had the genome, then you're really stuck, because the information needed to construct a cell in molecular detail from scratch is staggeringly large, although as of now, we don't yet know how to quantify it. Just as a little preview, for those of you interested in physics, how would you quantify the information needed to make a cell? It roughly amounts to asking, if I had a box roughly the size of a cell and I had all the correct atoms that I need to make a cell, what fraction of those atomic configurations would actually be living cells? As you can imagine, it's a tiny, 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 tiny fraction of configurations, and therefore it's a large amount of information. Okay, so what I've basically tried to convey to you is that in biology, yes, the genome is important. It contains information. The information metaphor is true. It's accurate. However, that information on its own is insufficient to make a living organism. What you need is a cell to read that information. And if you don't have a cell, then the information you need to make a cell is enormously large. And that resolves this paradox about the difference between, let's say, a movie about a mammoth and the mammoth itself. Okay, of course, you need a certain amount of information in the mammoth genome plus a mammoth cell to make a woolly mammoth. So ultimately, biology looks a little bit like other processes that humans use to make copies of things, right? You need an existing copy to make a copy. But that's a little misleading. Unlike a sculptor's mold or an architect's blueprint, biology doesn't use the existing copy of a cell as a template. Okay, it's not a photocopying machine. What biology does do is the information encoded in the genome is a recipe. It specifies a series of events that take place in space and time to create an object like a cake, right? But the twist, of course, is that you need a reader to read that recipe, a very specific reader, and that the thing that the recipe is encoding is itself the reader. And it's this sort of self-referential nature, it's this feedback that makes biology so interesting. And the person who came closest to articulating this is John von Neumann with his theory of self-replicating automata. And I think the ideas that he put forward decades ago still haven't been fully fleshed out and will form part of some of the great discoveries of the 21st century about how biology works. Thank you.